Yeah, I'm really glad that a lot of my stuff is not going to be, it's actually aligning really quite well with what we've heard already so far, so I'm quite happy about that. My name's Craig, I am from Flinders Uni. I work in a lab group called the Frontiers of Restoration Ecology, and I'm actually supporting a New Zealand-based People, Cities and Nature, which is an urban ecosystem restoration project funded out of the New Zealand government, and we're actually looking at the potential for health-promoting soil microbiomes. So it's a bit out there, but something a bit different. Yeah, healthy soils, healthy people. Can soil microbial diversity help boost immune and gut health? And yeah, probably similar to Tim, it's a team effort. I'm what you call an early career researcher, so I guess recently did a PhD in this area, but also have a background of nearly 20 years of working in soil data science, managing the soil information for South Australia. So my areas of research, there's a couple of areas of overlapping research. Soil microbiota, so that term microbiota is the microbial communities, and looking at those as indicators and facilitators of ecosystem restoration, and I'll talk about that a bit more. So I guess, yeah, this sort of topic is really from an ecosystem restoration focus, so putting more plants back into environments, or looking at the biodiversity of the plant-soil system. But hopefully what I'm going to talk about, there's some takeaway points for agricultural context as well. So, and then looking at overlaps of that with potential for beneficial contributions of those microbial communities to support human health, and that's mainly through the immune system. And I guess, yeah, microbes from the environment, they train, they educate, they prime, if you like, our immune system, perhaps also help regulate our immune system as we get older, but it's really important to set that up from a young age. So what is ecosystem restoration? Well, that's the process of assisting the recovery of an ecosystem that has been degraded. So there's kind of this concept of a restoration staircase where if you've got a really piece of degraded land, and it's all about what's the land used for, is it fit for purpose? So in that context, if you've got a post-mining scenario where it's, sort of, there's nothing really left of the soil profile there, or abandoned farmland that's being converted back to biodiversity values. So that context of having almost like a bare canvas or something in a poor state for its target land use, and building that up through various steps up to a target sort of land use at the end. And this context here is to help try and put back a native or more natural biodiverse plant soil system. So we've heard about the physical and chemical sort of lens that you look at soils, and really that's important in the early stages to look at your landforms in a post-mining context, I guess. Look at your landforms, look at the soil profile, the physical chemical structure, and then the next step is to really see how that biological aspect builds over time to support plant communities. And we heard from Tim about how, and others about how the microbial communities really support those ecosystem functions, and they do stuff to support plants. So yeah, this diagram here, we've got a sort of a time series of, and the diagram here is representing the complexity of a bacterial community, if you like, as it builds up in and around the root zone of a plant. So this is just an annual system, and you've got sort of initially perhaps fairly sparse but biodiverse bacteria in the soil, if you like, and over time the particular plant will push out exudates or sugars out from the roots. It'll attract and it will foster a relationship with a suite of microbes that are tailored to that plant. So each plant tends to favour its own type of community over time. And this is just an annual system, the example here. If you add more and more plants to that scenario, you get more interactions, more symbioses, if you like, more sort of interrelations, if you like. So we're talking about more diverse habitat being built for the microbes that live in the soil. And I'll get to why that's important for human health a bit later. Here's an example where we've got a grass and a legume or a nitrogen fixer. So they kind of, if you get more and more plants working together, they help each other out. So here the grass is producing factors which help with the rhizobium formation. The rhizobium help fix nitrogen in the legume, and then there's a bit of excess nitrogen, and that can go back to the grass. So more and more plants, more diversity, there's more potential beneficial relations happening there. I'll talk a little bit about soil microbes and nutrients. Some of this stuff was really new to me reading into this. So what I should have said is I'm going to present some of the literature and then I'll get into some of our own work, all right? So just to give you a bit of a wide scan of this area. So some of this stuff I found really interesting, learning this stuff over the last few years. So if I start on the right, and this is kind of a story that we've kind of already heard today, where if you've got excess nutrients in the soil, the plants will lose that incentive for root growth. And they'll lose incentive to sort of foster relationships with the fungal, sort of the mycorrhizal fungi. So too much nutrients will, I guess, make the microbes a bit lazy. So you're not fostering those relationships, whereas if there's kind of the right amount of nutrients, the plants and the roots and the mycorrhizal fungi are working as they should, I guess. So these other papers here, what we're talking about here is that plants not only take up nitrogen in this case, as an example, they don't only take it up in the soluble, inorganic form, so whether it's ammonia or nitrate, they don't only take it up in that form, they also actually take up nitrogen in the form of breakdown products of microbes. So they take it up in amino acids, peptides, proteins, and actually whole bacteria are kind of consumed by the plant roots. So there's kind of this concept of rhizophagy, where a plant root will basically munch on bacteria. This is a concept that's now, I guess, recently in the literature. And so what we're doing, this is a study looking in a post-mining context that I did recently, and this is a measure we've got time, obviously, on the x-axis, and we've got sort of, on the y-axis here, this is a measure of the whole community of a bacteria, of the bacterial communities in a post-mining context. So you kind of look at this through what they call a restoration chronosequence, which is basically different ages of the vegetation that is growing in a post-mining context. So, and this is looking at, and it's basically saying that the community of the bacteria shifts over time as you put more and more diversity back into that landscape. So starting off with something that's, quite young, to more years, and then the remnant. And remnants have a lot of diversity. So the bacterial diversity in a native system is going to differ among different native systems. But this measure basically looked at the changes in that whole profile of that community and how similar it was over time to approach what was the target in this case of being a biodiverse system. And within this mining scenario where it was sort of reduced back to a bare minimum, it was going to take 40 to 60 years for the bacterial communities to recover, basically to what would be considered something similar to a reference state. So yeah, getting now to the human health angle. So the World Health Organization, in a 2015 sort of state of knowledge review, they pulled together a bunch of information on how biodiversity helps support human health. And they highlighted microbial diversity as one of those branches, if you like, of how biodiversity through microbial diversity can help support human health. It does this through supporting the training of your immune system, and particularly from a young age, as I said. So in this slide here, we've got a bit of information, but kind of what I want to impress on you is that as humans, we're not standalone organisms. We're actually a system where we've got a whole bunch of microbial communities in us and on us that perform important functions. So we're not a standalone organism. And the genetic capacity or the ability to do stuff from the microbes that are in us and on us, almost studies suggest that outweighs what our own genetic capacity can do as the human genes. There's the microbes, they just are so important, for our gut in particular, as an example. So, and there's a lot of medical literature. Increasingly, they're finding associations between the microbes and various different diseases and health status. So different factors help shape our microbiome. So how you're born, whether or not you're breastfed, your diet is critical, but also the environment that you live in is critical to the microbes that kind of get on you, land on you, get in your airway, and work your way into the gut. So going back a few years, this sort of reason that we're increasingly disconnected from natural sources of microbial diversity. The thinking has been around for a while, and soils are a particularly rich source of microbial diversity in the environment. There's been increasing allergies and autoimmune diseases, and this kind of idea that maybe disconnection from natural sources of microbial diversity could be part of the reason for that. And it's kind of gaining in support in recent years. Also, we're finding that the green spaces or the types of bacterial communities or microbial communities that live in those green spaces in urban areas, they're kind of becoming a bit the same. It's kind of the same types of plant communities, the same type of management. So globally, in urban green spaces, we're kind of surrounded by soils that are a bit kind of samey. And we saw the slide earlier about how urban populations are just sort of taking off. All right, so this slide here, what we've got is what I'm going to talk about is how microbes from the environment can come in and help boost our immune health. So 4 different ways that microbes can do that. And the slide here is we've got cells that line the gut wall. Okay, so microbes can come in, perhaps we breathe them in, they can work their way to be swallowed and they get in the gut. So they can kind of add to the protective communities that line our gut wall. Not all of them, some of them are suited, a lot perhaps aren't. They can also actively participate in immune signalling. So here we've got an immune cell that's reaching through the cells of the gut wall. It's sampling this bacteria. There's a molecular pattern in that bacteria that's saying something, and then it can go and trigger either defensive inflammation or it can actually trigger tolerance. It can trigger anti-inflammatory responses. We're obviously building immune memory and antibodies. And the other sort of functional capacity that it can do, they can come in and they can add, I guess, functional capacity to what those microbes are doing for us in our gut, breaking down organic matter, producing metabolites or breakdown products, such as short-chain fatty acids, and perhaps I'll go into one of them, what that means later. So I guess I want you to think about the coevolution that's happened between microbes and humans, biodiversity, this concept of diversity or biodiversity being really important, but also this concept of old friends, which is that perhaps there's key species that perform really important roles. So both diversity and key species. That diversity helps counteract pathogenicity. Probably touched on that a bit earlier. We've got a biological connection between our environment and our own health, and it can impact the whole body, particularly through the gut, and shape the course of infectious and non-infectious diseases. Okay, so this is I guess a hypothetical dose-response curve, talking about a particular key species, let's say E. coli, all right? That's probably got a bad rap, but there's lots of E. coli doing, pretty good stuff for us most of the time. Pick a particular bacteria. So we've got low levels of it, exposure to it, and very high levels, and here we're nice and healthy and happy, here we're sick. Okay, so this is a hypothetical dose-response curve for in a hospital environment, you've got sick, immune-compromised people. The message here is the only good microbe is a dead microbe, okay? Let's keep everything super clean. And this is kind of the message, rightly, perhaps through COVID, everyone's super clean with the hand wipes and all that. So good for hospitals, good for when you've got sick people around. But is that somewhere, is that thinking, should that be applied everywhere all the time? So perhaps we should be thinking something more like this, where, yes, very, very high doses of exposure to a bacteria, the bacteria start talking to each other. There's a thing called quorum sensing where they go, hey, there's heaps of us here, let's have a party, okay? So infections occur when you've got too much of something, but at this end, where there's not enough exposure to a key species, the immune system is not getting the training that it once would have got that we're used to from an evolutionary sense. Whereas in the middle, perhaps at some low dose of a key species, where it's kind of got the protective action of biodiversity to keep it under control, that is perhaps where, from an evolutionary point we were, and perhaps that's something that we should look to returning to in the general sense. Okay, has everyone had their lunch? So this is just going to highlight this point a bit. This is an image of a traditional African hunter who's gone out and got his free-range organic zebra for lunch. He's kind of prepared the zebra meal for lunch, and now he's washing his hands in the microbial-rich gut contents of the zebra. So pretty gross, I'm not advocating this, but I guess we're building immune fitness, and we've got diversity that's controlling pathogenicity. So just trying to drive this home. This is well away from our modern concept of hygiene. Not suggesting anyone do this, but, are we too clean? Some further sort of ideas in this space. when you have poor diet and lifestyle and too much stress, not enough sleep, too much antibiotics, you can lose key species in your gut, and that's when problems can occur. But there's also this finding that the bacteria in your gut are dominated by spore formers. So they do things in an anaerobic or very low oxygen, no oxygen state, but they can form this protective spore because they come and go and spend time in the environment. So the fact that your bacteria in your gut are dominated by spore formers makes sense that they spend time in the outside environment and perhaps do things in the environment and then work their way back in, come in on food perhaps, or in the air. So we have this kind of idea that there's two layers of protective biodiversity from the microbial point of view in our gut, in our microbiome of our gut, skin, airway, and also this outer layer in the environment of a second layer of protective microbial diversity that can then come and supplement us if we are lacking. So studies have found that being out in the environment, you can get the bacteria, for example, get blown up in the wind from soils as a big source. These can be detected and settle on you, on your skin, in your airway within as little as 15 minutes. And they do actually associate vertically with closeness to the soil, but also in response to what the vegetation type and is around you. So the aerobiome, or the microbial communities in the air, is associated with your environment, and you do get exposed to that. It does kind of land on you and work its way into you. And as I said, we're systems, we're not standalone organisms. A couple of studies here. The left one is showing that children growing up on farms have lower rates of asthma and allergy, and that kind of, anecdotally, that kind of makes sense, or you kind of hear that a lot. The study on the right shows that the more hands-on or traditional farming practices with greater exposure to soil and manures, they have even lower rates of asthma and allergy than the more industrial sort of farming techniques. With mice studies, because you can control a lot of things in mice, adding soil into there, it changes their gut microbiome, and it triggers more anti-inflammatory immune responses. A couple of other mouse studies here, so exposure to soil dust, accelerates recovery from the antibiotic sort of treatments because that can tend to wipe the slate clean. If you've got no community there, it's ripe for something else to come in. Here, they've kind of found with a lot of these mouse studies that they're almost too clean, and they're a bit worried about that the immune system of some of these super clean mice is not a natural sort of immune system, and they need to do that to control all the various things that you need to control in experiments. But they've started looking at more exposing mice to some controlled levels of germs just to activate their immune system a bit more. Here's one that shows that exposure to soil and plant materials helps reduce the incidence of colorectal cancers. Okay, but yeah, soils are not all the same, and I'll kind of get to this in a sec. Here was a study in Finland where basically they brought in soil and plant materials into the daycare yards, and the kids had better immune responses within about a month. They had improved biomarkers for sort of anti-inflammatory immune responses compared to concrete and fake lawn. So soil microbial diversity on its own, and that is driven by plants, as I said, but on its own it can help control both bacterial pathogens and antibiotic resistance. There's a lot of talk about antibiotic resistance genes in the environment, but microbial diversity can help counteract the build-up and help slow them down over time. This is a study that I did as part of my PhD, looking spatially across regional Australia in local government areas. So on the y-axis, we've got different socioeconomic classes. On the x-axis. On the y-axis, we've got, this is levels of hospitalizations for infectious and parasitic disease risk. And if you take just the lower socioeconomic class, which is kind of where you expect to see environmental health sort of impacts fall out, what it's sort of showing here, I was particularly interested in the measure of soil that was available in the environment, and to try and relate it to microbial diversity. And I used a measure of soil cation exchange capacity, which is a bit technical, but it's kind of relating to organic matter and clay content, and we've heard about them. Organic matter and clay content both provide more habitat for microbes. So in areas with poorer quality soils, so less organic matter, less clay content, typically lower microbial diversity, they had higher rates of infectious and parasitic diseases. So perhaps they're not getting the level of immune training that some of these other areas are that have more organic matter and more microbial diversity in their soils. This is digging into some data from just down the road at Mount Bold Reservoir Reserve, where there was another one of those restoration chronosequences where there was abandoned farmland, I guess, to remnant sort of patches. So it's about the biodiversity, a gradient of biodiversity in the plant soil system. And I was interested to look at what sort of bacteria were favoured at this end versus what sort of bacteria were favoured at this end. And the simple kind of message here was that, and we're not talking, so the simple message was that as you feed your soil simple and unreliable food, you're kind of creating, you're favouring more of those pioneer species, so fast-growing, generalists, opportunists, and not to say that they are pathogens, but they've got more of those potential pathogenic traits because opportunistic, fast-growing generalists, they can just go for it potentially, whereas here, these were slower, the ones that were particularly favoured were slower growing and suited to more complex sort of food sources. So different types of bacterial communities are favoured at either end of that gradient of biodiversity in the plant soil system. Because we're looking at health, we thought, we should do a mouse study. We won't be taken seriously unless we do a mouse study. So we had some lab mice in a old school sort of, these days the mice are all in little air-conditioned units by themselves. We had an old school open top wire cage in a big rack that we kind of wrapped in plastic with vents so they didn't suffocate, and we had sort of trays of soil and a little USB desk fan here, and we exposed because we wanted to test exposure of the mice and what happens in their gut to a realistic exposure to soil, because a lot of the studies are, they force feed it or they inject something or whatever. So we wanted to test the impact to see if there was a change in the gut, really, from a realistic exposure to just low levels of dust. So we had no soil at all. We had low biodiversity soil from the Mount Bold Reservoir Reserve, actually, a fire break with minimal sort of plant diversity, and it was low bacterial diversity. And then we had high biodiversity, which was high, from the remnant site, high plant diversity and high bacterial diversity. And the message to come from that was that there was a small, but over 7 weeks we exposed these mice, there was a small but significant change in the overall composition of the mouse gut bacterial community, so it did make a difference. And we also we tested for anti-anxiety-like behaviour. So the way that you test for anxiety-like behaviour in mice, one of the ways is you can put them in a little open-top box. You have a video tracking thing, and you time the time spent in the centre and time spent in the corners or the sides. And I looked at what changed in the composition, what bacteria changed in the gut between the start and the end. And there was a whole bunch of mouse intestinal bacteria, and that was a bit boring. But there was one particular bacteria that had been found in a soil environment. It was a spore former. It was anaerobic, so it was kind of doing things in an anaerobic environment. It was spore forming, so it could come and go. And it was butyrate producing. And I looked up, what's butyrate? And butyrate is a breakdown product. It's a fermentative breakdown product of organic matter. And it's like a really key thing for gut health, because basically the cells that line your gut, they need butyrate to live. And this particular bacteria was correlated with less anxious behaviour in the most anxious mice. So there was kind of like a link there that was like, something going on here. It's kind of preliminary and being looked into further, because with any of these things, you're kind of opening up new questions, and every study has limitations. But basically, we've kind of this thought that there might be bacteria that are breaking down organic matter, and you break down organic matter in your gut, you break down organic matter in soils. Occasionally, those soils get quite wet, and these bacteria can form spores and basically cruise around the place. So this idea that maybe there's beneficial bacteria that have a home in the environment and a home in the gut, and they can be performing beneficial roles. And there's probably perhaps one of many different mechanisms through which microbial diversity can help us out. We've got a PhD student sort of looking into this further. So last slide. So these ideas are being taken seriously, I guess, in the medical arena. So this was a paper that one guy by the name of Jake Robinson in our lab group. We got it into Lancet Planetary Health, which is kind of a cool medical journal, but the idea that ecosystem restoration is integral to humanity's recovery from COVID-19 as a bit of a case study, but among a number of different mechanisms, I guess pushing the idea that restoring plant soil systems, restoring microbial diversity can help build immune health. So that's all I've got.